Hey everyone, welcome to a new episode of the AI-Based Business Show, the show where you will learn about tech, entrepreneurship, business, money, and just anything that's related to the money world. Today we have a special episode, special guest, an industry that's dear to my heart that I believe everyone should be paying attention to because it's an industry that could change how we as human lives on Earth, and that's the biotech industry. Andy is a builder, is a researcher, is a scientist, is an entrepreneur in that space. And today I have with me Andy on the show. Andy, welcome.
Thank you, Ahmad. Great to be here. I'm excited for the conversation.
I'm really excited to the conversation. Just a note for the audience, I'm not related to the biotech industry. I may be a noob in some of the areas I'm going to ask questions about. So if I've asked questions that some of the listeners might be experienced to, excuse my kind of ignorance on the industry, but it's a great opportunity to learn. Uh, Andy, you have an amazing journey. You got into biotech, molecular diagnostics. Take us through kind of your multiple exits, your career. How did you end up here? How can a kind of researcher become an entrepreneur and build companies?
Yeah, great. Thanks, Ahmad. And again, thank you for the opportunity to share my story and the company that we're working on right now. Before co-founding our company, Inherent Biosciences, I led commercialization in both large enterprises, large biotech companies, and then also in startup biotech companies. And the largest was Roche Diagnostics, and then a number of biotech startups, and have gone through exits with those companies. And my background when I was studying, in my undergraduate education, I was really fascinated with molecular biology. And at the time too, there was, a lot of news and things happening around the Human Genome Project and getting the entire human genome sequenced. And there was a lot of hype and excitement about that. And I certainly caught the wave of excitement there. And at one point, I was trying to decide, as I was getting ready to graduate, if I should go to do a PhD program or go another direction, like go into a biotech company. And ultimately, I decided to go into a biotech company, and instead of going like an academic researcher PhD kind of program. And that was really transformational for me. The experience that I had initially in that startup biotech company in Texas was really cool, fun, exciting, and it whetted my appetite for that kind of environment, fast-paced, and a lot of learning. And so, but I just had, at the time, I just had a bachelor's degree in molecular biology, and I was working at the bench doing lab work. And I realized very quickly that I needed to either go get a PhD or I needed to move to the business side
Right.
and learn more about commercial things. And so that's when I went in that direction.
What's for a coming up biotech student or a person who had your same background? How did you make that decision?
Yeah, it was pretty easy because it was a little bit of a financial decision at the
Okay.
time. If I went into the academic route, the financial economics was, you know, essentially being poor for the next 5 to 7 years
Yeah.
to get a PhD. I also had an opportunity to work in a laboratory for an academic, really amazing scientist. But he was going to pay me less than if I went into the biotech company. If I went into the biotech company, I'd make a few $1,000s more a year. And at the time, having just graduated, that was attractive enough that it helped me decide to go into the biotech company. And I'm really glad that I did.
That's amazing because I have my fiance. She just graduated biotech. She did genetics research on cancer with her master's degree, and she's always debating whether she wants to go into academia or go into like a biotech company. And I'm like, your choice should be a biotech company because yes, you go into academia, you work in a lab and you do all this research and you write these papers. But when you get into like a biotech company, you could take what you're working on in the lab and commercialize it and give it to people and advance and get in the
Yeah.
hands of people. And I just like love this concept.
Yeah. Yeah. And I think too, it depends on what that end result that you want to achieve. Like if she wants to teach and be a basic science researcher, then that academic track might be a good fit for her. For me, I needed a lot more impact, and I wanted to, and you can still have impact, certainly teaching
Right.
and doing basic research, but I wanted to see the translation of technology having a direct impact, and in my case, in patients. And I think that's one of the key things is what do you want to accomplish, and then what's the best path to get to that. My dad was a physician, and so I grew up in kind of a healthcare environment, and I was really attuned to making a difference for patients and bringing a little bit better life experience to people. And so that kind of helped me decide which way to go.
Okay. You mentioned in your talk that you worked on commercializing a bunch of molecular DNA based diagnostics in
Yeah.
5 different startups. Uh, I want to understand, how does the commercialization process go? What are the
Yeah.
challenges that we have in the market, and what can we bring as entrepreneurs to make this process more innovative?
Yeah. Yeah. So molecular diagnostics, that just means that you're using a molecular based approach to understanding what's happening, what's going on. And that can be DNA, it can be other molecules that you look at as well. And some of the biggest challenges is that the development process for a molecular diagnostic is not that far different from a therapeutic. I
Okay.
mean, there's a lot of similar steps that you have to go through, and the clinical utility, is it safe and effective? It does it actually do what you say it does? Is it accurate? Does it have good performance in diagnosing? And yet, the valuation and the investment kind of outcome for a molecular diagnostic tends to be much lower than a therapeutic. And so that's a big challenge. And I think that when you look at investor appetite for a molecular diagnostic versus a therapeutic, a lot of investors will only invest in a therapeutic. It's a higher risk reward ratio. A molecular diagnostic, though, if you don't have good diagnostics, you often are doing trial and error medicine on
Okay.
the therapeutic side. You're saying, well, we don't know exactly what's going on here, but let's try this therapeutic and we'll see how it works. And when that doesn't work after 3 or 6 months, it's like, oh, well, let's switch to this other therapeutic. So there's a tremendous amount of trial and error, medicine in our healthcare, that is pretty frustrating, and it's disappointing, and it's hard for patients. My mother had severe rheumatoid arthritis, and I watched her go through this.
Right.
I watched her go from one therapy to another therapy, and not really seeing an improvement in her symptoms. And so I just kind of, my perspective is like, there's got to be a better way for us to do this. And there's some challenges with regulatory as well. I mean, just like with the therapeutic, there's a regulatory body, the FDA, that says, okay,
Right.
if you're going to put a medical device into the market, it has to be safe and effective, and we need to approve it before you do that. But there's some unique ways that we have been able to bring molecular diagnostics into the market more quickly and have an impact for patients much sooner, and also cost effectively. And I'm happy to talk about those regulatory kind of breakthroughs that have helped with the ability to do that for molecular diagnostics.
Could you mention some?
Yeah, yeah. So in if you're bringing to market a medical device,
Mhm.
and that can be an assay, then the FDA has a process called a 510K approval or a pre-market approval. And 510K is for the part of the Federal Drug and Cosmetics Act, and it's Section 510K. And so, and in the past, if you had a molecular diagnostic, there's a couple ways you can bring that to market. So you can have it a system that you sell to a physician office that they use to then test somebody and do the diagnostic process and see what's going on. The other way is what's called a send out test. So there's a specimen collection that happens either with the physician or the patient directly, and then it's sent to a lab like LabCorp or Quest, and they do the processing of that specimen and provide a result. Well, there's two main regulatory organizations that regulate clinical laboratories like LabCorp and Quest, and it's called CAP, which stands for the College of American Pathology, and CLIA. And that CLIA is the federal government, and it's the Clinical Laboratory Improvement Acts or amendments, Clinical Laboratory Improvement Amendments. And these two organizations regulate clinical laboratories, and they enable laboratories to clinically validate a molecular diagnostic and provide that as what's called a lab developed test. So a lab developed test is there's actually information about this on the FDA website. It says a laboratory developed test, or an LDT, is a type of in vitro diagnostic test that is designed, manufactured, and used within a single laboratory. So you have it in the laboratory, you validate it, and then you can market that as a clinical diagnostic, and it's regulated by CLIA and by CAP. And you can have both or just one. If you could just have a CLIA validated assay. And so that enables us to get a test into the market more quickly than a traditional medical device 510K FDA approval process. And it's still regulated at a very high level because those organizations come in and inspect the laboratory and confirm that you're doing things according to the policies and procedures that are in place, and so that it has good accuracy, good performance, and safe and effective. And that's been a really great benefit. The FDA has talked about potentially regulating lab developed tests, and that remains to be seen if that's going to happen. And if it does, then that will add a pretty big extra burden on molecular diagnostics. It would be a big impact in the industry if that happened.
Okay. It's always a debate whether regulatory is a great thing for the industry. Of course it is, or it's not. And after I watched Poisoned on Netflix, I don't know if you watched it yet, but it speaks about all the different epidemics that happen kind of in the food industry and where the regulation is missing. I'm like, okay, now I'm in this debate. Is regulation good? Is it bad? Should we have reduce it? And it's a whole debate we could spend hours talking about. But I want to jump in what you're doing right now, inherent bioscience. How did it come into existence? If I was a fly on the wall when you were sitting and thinking about inherent bioscience, give me an eye look of your inspiration behind it, how it started, and what do you guys do?
Yeah, thank you. So the last exit that I went through, after that happened, I started thinking about what's the next clinical problems that need to be solved. And I have some friends who were going through infertility treatment, and they had been trying to get pregnant for over 2 years. They spent about $80,000 US mostly out of pocket.
Wow.
And then their physician had diagnosed them with unexplained infertility. And that's what caught my attention. I was like, okay, wait, like, why is this unexplained. How often does this happen? Where's the gaps, that need to be filled? And I was surprised to learn at the time it was 1 in 8 people experience infertility. Now, in the last 4 years, it's grown to 1 in 6,
Wow.
according to the World Health Organization. So it's an increasing problem, and a large percentage of people are diagnosed with unexplained infertility. It's a significant number, and that's when you see that, especially as a molecular diagnostics background, it's like there's an opportunity there. There's something missing, and so I saw that there's a lot of focus on the female partner. She gets extensive and invasive testing and treatment, and yet up to 50% is due to male factors.
Partner. Right.
Yeah, and when I dug into that, the standard of care for assessing the male factor is like 6th grade science.
Right.
It's using a microscope to count how many sperm are there and how many are swimming. And that's when it was like, oh my gosh, there's got to be a better way for us to assess,
a deeper, like, the problem. I want to ask a quick question here. You mentioned that after your last exit, you were sitting there to see what's the next clinical problem that is there to solve. How do you, like, how does a person in that field look at, like, find out what are these problems or decide on a problem? Like, what is the criteria to choose, or where do you find these problems?
Yeah, so there's I think, and you're asking a great question, Ahmad, because one approach is you can talk to the physicians and you can ask. In this case, it was patients, and I think there's a lot of value in starting with patients and understanding what's the patient journey. What is it that the patient is going through? Like seeing my mother go through her healthcare journey for severe rheumatoid arthritis, and then my friends going through infertility. And then once you know what's happening with the patient and the problems that the patient is facing, then you can start to go into, well, who are the physicians that are working with these patients, and how are they doing that? And then what are the challenges and the problems that they face in taking care of these patients? And by focusing on the problem and really understanding it and diving deep on the problem, then often the solution emerges. And so when there's a problem like this, where it's a diagnostic problem and it's unexplained and we don't know what's going on, and then you see, oh, you're using like 80-year-old technology to try and understand what's happening. I mean, you're using a microscope to count sperm. That's
doesn't work.
The state of, I mean, there's an opportunity right there, and my background had been with DNA sequence. So looking at the sequence of the DNA, and it's so how, people have tried to look at the sequence of DNA and sperm and tried to come up with tools for this problem, and it hasn't really been a lot. There hasn't been a lot of, really robust things around the DNA sequence. So there's got to be something more than DNA sequence, and that's where. And then, we have an amazing team at our company, and the co-founder of our company, Dr. Kristen Brogard, who's a PhD in this field, I was really fortunate to get introduced to her a little over 4 years ago. And we would not exist without the team that has come together. And that's not just, everybody who's full-time, but investors and collaborators and clinicians, and then everybody who has contributed to what we're doing. Now we just got a grant from the NIH to help fund some of our product development, and without that, we wouldn't exist. We wouldn't have made the progress and have the tools and the products that we've been able to get available to physicians and to patients without a lot of people involved.
I love this approach because a lot of people look at biotech and the whole kind of bioscience industry as an industry that's super in the lab, in the research. But what you just said is like your thinking is just like any entrepreneur. When you find a startup, you're out in the world and you find a problem that there's no solution to, but many people encounter, and you go and
Yeah.
build a product to solve that problem. You did
Yeah.
the same. You found people who have a problem that's not
Yeah.
being solved. You went back and sourced why isn't that solved, and you're building a solution for that problem. And
Yeah.
Lynn Kawas, one of your investors, and
Investors.
she was on the pod. She mentioned something really interesting when I was asking about this topic. Is like, what do you advise entrepreneurs in the biotech industry? And she's like, once they start their research, they should be thinking about their commercialization
Yeah.
aspect and how they're going to do that. And that helps them think how the problem helps people, like what's the solution and how it helps
So,
people, and they go back to solving the problem. And that's an
yeah.
amazing way to think about it.
Yeah. Yeah. I think it's critical. It's there's a lot of examples of people who have developed solutions, products with amazing technology, but it's not really solving a problem. It's just
Yes,
like, you
it's
know,
just there.
yeah. And the question that like, it's the so what? Like, okay, this is really amazing technology, but so what? Like, how does
Right.
this actually solve a
Okay.
problem that is really meaningful? Meaning that if someone exchanges money with you, the
What are they exchanging
value,
for?
yeah, the value that they're getting out of the solution you're providing
Okay.
to solve the problem that is a
Okay.
lot of pain for them
Right.
is more than how much money they're parting with,
Yeah.
to get the solution. So, yeah.
Therapeutics, epigenetics. Can you give me a quick definition to both of these
Yeah.
terms?
Yeah. So you mentioned epigenetics. So I talked about the DNA sequence,
Right.
and the Human Genome Project
Okay.
was to sequence all the DNA. And everybody thought, oh my gosh, when we have the human genome sequenced and we can sequence our whole genome,
Yeah.
then we're going to have precision and personalized medicine, and it's going to change everything in healthcare. And really, for the most part, that hasn't. I mean, it's been like 20 years since the
And
Human
hasn't happened.
Genome Project. It hasn't really happened. There's been a few cases where the DNA sequence makes a huge difference. Single gene diseases that it's a specific mutation, or cancer where there's a accumulation of mutations. But for the most part, when you look at complex disease or chronic disease, the increasing prevalence of those diseases is not explained by sequence variation of the DNA. So there has to be something more going on. And the field that has been emerging is called epigenetics. One of the fields that's been emerging. And epi is a prefix that means above or on top of, and then genetics is the DNA sequence. So epigenetics is what's on top of the DNA that's turning on and off gene expression. And there's a lot of rich information about that. So that's epigenetics. And the the thing about epigenetics is that it is independent of the sequence, and it's influenced by our environment.
Okay.
And the cells that all of our cells have the same DNA sequence, but they have different epigenetics. It's kind of like the DNA sequence in our cells is like the hardware.
Right.
The epigenetics is the software.
Right.
And there's different software for your skin cell and for your eye cell, for your brain cell. That software is different, and that's the epigenetics. And so that's what we look at is in our case in sperm, the epigenetics is very unique in sperm, and it's turning on and off different genes that are important for the sperm to be a sperm and to function really well and to be able to do its job. And in that software for the sperm, the biological software, there can be bugs. There can be bugs in the
Okay.
software,
Okay.
that can make it so that that sperm doesn't function very well. And that's the kind of technology that we've been able to leverage in providing insights that haven't existed before in the clinic anyway, about what's going on with this patient's sperm, and then how do we help them get pregnant and have a baby.
Okay, that's a good path to speak about Path SpermQT, which is your product
Yeah.
at Inherent Bioscience. Um,
Yeah.
you mentioned you help these patients get insights on why they're not being able to get pregnant
Yeah.
studying that sperm. What insights are you giving? How
Yeah.
is this insight happening? Take me through the process of like really what's happening behind the
Yeah.
closed doors where you're able to give them that
Okay.
diagnostic.
Okay. Great question. Yeah. So when we set out to solve this problem, or at least provide a better tool to understand what's happening, we took a different approach than everybody else that had been doing this. Typically, what people have done is they take a group of patients that have the problem,
Okay.
and then they look and see what specifically is different in those patients. Like what's the thing that's different in the epigenetics specifically with those patients. And we took the opposite approach. So we said, what does really good, healthy, normal, high functioning sperm look like
Okay.
epigenetically at the molecular level? And then any deviation from that would not be good. It's kind of like when you go to the doctor and they check out your heart
Right.
and you get like an electrocardiogram.
Right.
When they do that, they either tell you have a normal electrocardiogram or abnormal.
Right.
And abnormal can be a whole bunch of different things. And so we did the same thing. It's like, what does normal look like?
Okay.
And then abnormal can be a whole bunch of different things. And so we looked at the sperm epigenetics, and we found our hypothesis was that for a sperm to be a sperm, it has tight regulation and control over the parts of the epigenome, or that are most important. So the genes and the epigenetics on those genes that are most important. And then if those deviate from that, then it's not good. So we found 1,233 gene promoters.
Wow.
It's the part of that transcribes the gene and makes a protein. We found 1,233 that are really important for sperm. And there are multiple cellular pathways. And so we analyze that. When the patient goes to a fertility doctor and they start this process, this journey of finding out what's going on, then the physician orders our test, and we send a specimen collection kit to the patient. They collect the specimen in the privacy of their own home, and then they ship it to our laboratory, and we break open the head of the sperm, analyze the DNA, what's on top of the DNA, the epigenetics, and we look at these 1,233 promoters, and we can see if there's what we call dysregulation, that it's just being, it's dysregulated, and we can see that. And then when we started looking at this, we didn't know what the data would show.
Right.
And what we found was that, typically people start with artificial insemination. It's a procedure where they take sperm and put it close to where the egg is in the uterus so that the fertilization can happen. And that's usually the first procedure. There's another procedure called in vitro fertilization, where they take an egg out of the woman and they inject, or they put the sperm with the egg outside in vitro, and then fertilize the egg and then transfer that after it grows into an embryo. And what we found in our data was that the procedure of artificial insemination is failing at a much higher rate if you have a problem with the sperm epigenetics.
Okay.
But if you go to in vitro fertilization, it works.
Right.
And so what it's looking at really from a molecular standpoint is the ability of the sperm to find the egg, bind to the egg, penetrate the egg, and fertilize the egg.
Okay.
And if it has an epigenetic abnormality there, it can't do that very well, and it needs more help. And so it needs to go to in vitro fertilization, and there's a procedure that they do called ICSI. So it's IVF, in vitro fertilization with ICSI. ICSI is ICSI, and it stands for intracytoplasmic sperm injection.
Okay.
So this is where a highly trained technician with and they get a microscope and they take one egg from the female partner, and they pick one sperm, and they inject the sperm into the egg. They do the job of the sperm for it.
Okay.
And it's a really expensive procedure. It's invasive, and it's usually later in the fertility journey. But if there's a male factor, if there's a problem with the sperm, this procedure works a lot of the time. And so what we can do with our test is we can identify. So what happens if you have 100 guys coming into the fertility clinic with their partner and they're trying to get pregnant, and if you only look at the sperm with a microscope and count how many are there and how many are swimming, you're going to find like 5 or 10 or so that have a low sperm count, and they're going to need to do IVF with ICSI. We're finding an additional 20 men.
Okay.
So it's a significant increase in diagnostic yield. And those men will be better off going to IVF with ICSI instead of starting with artificial insemination and doing cycles that aren't going to work.
So kind of what you're doing, the process for IVF and artificial insemination is already there. It exists in the real world. It's a solution that exists and that works.
Yeah.
Artificial insemination has been proven to work less and where it's a sperm problem. So we tend to go down to the IVF part, and then if the IVF part has a problem, we tend to go to the IVF ICSI part, right?
If the IVF, the ICSI part, you got it exactly.
Okay.
Yeah.
So coming down there, we have the solution that works. What you guys are doing at Inherent Bioscience through SpermQT is identifying the more male partners that are having this problem, that using regular testing are being identified.
You got it. That's perfect. Exactly right. So these men are going undiagnosed.
Right.
They get a semen analysis. They count how many sperm are there and how many are swimming, and they're like, "You look good, plenty of sperm, and they can swim." Okay. And there's 20 to 25% of those men that have an epigenetic issue with their sperm. And so when those men go undiagnosed, then women are put through treatment and procedures that's not going to work.
Okay, so basically the problem today is when you do a regular sperm analysis or sperm test, is you're looking at how many of these sperms work, and you're like, okay, this works, without looking at the epigenetics, and this is where the problem is, and this is what you guys are doing.
That's exactly right. Yeah.
Amazing.
Yeah, the men go undiagnosed, and that's why we're explaining, a portion of that unexplained infertility.
Okay. You did that test, you found this addition in men. What's the next step after that?
Yeah.
What comes after?
So the physician then uses this information to counsel those patients to go, we can do artificial insemination, but your success rate is significantly lower than the normal population.
Okay.
If we do IVF with ICSI, then you have the same success rate as the normal population. So it's guiding treatment between those two things. And then also they can do intervention to improve the quality and the epigenetics of sperm. And so that's just a new area that we're starting to uncover, because epigenetics is influenced by our environment, what we eat, our stress. the different toxins that we're exposed to, all of that can impact the epigenetics. And so if you change that, it can also change the epigenetics of the sperm and improve that sperm quality, and it can have they can get better outcomes that way.
Okay. So what you're saying is we may in the future get a startup or someone who might go start a company where he works on fixing these epigenetic problems using the natural way instead of going through an IVF and all these stuff. Like would that, like if we got a human personalized medicine and I got to change that epigenetic through natural causes, what I eat, my environment, and all that, would I be able to solve the problem without going to an IVF, or is an IVF would still be necessary?
Yeah, no, so we have case examples of men who have done what the doctor told them they needed to do. So you go to the doctor and they say, okay, you need to make these changes to improve your chances of having success and improving your sperm. And unfortunately, a lot of men don't do those things, but some do, and they really lean into that. And so, and we've had situations where we've tested them and they had a poor epigenetic sperm quality, and then they made these changes and we tested them again and they moved into the normal. They look like the normal range, like the normal population. And so that gives us, some hope that there's opportunities to do that. And then the other area is therapeutics. And you asked about therapeutics. So a therapy can be something like this, a lifestyle change. That's a therapeutic intervention. There can also be drugs that can be provided that can also make a change in the epigenetics. And I'll give you a specific example. So there's a condition with some men where there's no sperm in their semen. Okay. And this is called azoospermia. And when this happens, they are referred to a reproductive urologist, a specialist, to help these guys see if they can get sperm so that they can conceive with their own sperm. And if they can't, then they have to go get donor sperm from a donor. So there's one of the ways that they do this is through what's called a testicular sperm extraction. Okay. And the reproductive urologist fillets open the testicle and searches for sperm inside the testicle.
Oh, wow. Okay.
Yeah, it's very invasive and it's very expensive. It's usually not covered by insurance.
Wow.
This is a elective procedure. It's quite expensive. And it's, you can imagine going through that. And about half the time, they can't find any sperm. And so the first question is, why does this happen? Like, what's causing it? Well, one of the academic collaborators that we work with on this product development and these areas identified a specific epi mutation in a cellular pathway that results in this abnormality in spermatogenesis, that's where you make sperm, and so it's not working right, and it's in a similar cellular pathway as another disease called Crohn's disease. So it's like a mismatch DNA repair pathway, and in a cell model, she showed how she could reverse this, and the cells would start producing the protein by taking a CRISPR, which you may have heard of. CRISPR targets a specific location in the genome, and then it can change the sequence. The difference was you take CRISPR and you fuse it to a methylase activity. So it's a methyl group. It's changing the methyl group on top of the DNA. That's the epigenetics that we look at.
Wow.
And so in cells, she showed this potential therapeutic intervention can rescue these cells that aren't producing this protein for, and it's causing like azoospermia. And so that could be a potential therapeutic that, and it explains, so this epi mutation in our initial data with the academic collaborator, it explains about 18% of these cases for patients. And that's a pretty good portion of it.
Right.
So those therapeutics, I think, are on the horizon. It's going to take some time because, as we know, the therapeutic development timeline is quite extensive, and to be able to get safe and effective treatment approved is gonna take a while. But we think that this is, the future in a lot of medicine is going to be centered around epigenetics versus like a specific gene sequence being changed.
Okay,
things like
you're
that.
looking at the outside of the cell and then figure out the problem.
Yeah.
How would the epigenetic shape personalized medicine?
Yeah, I think it's a there's a great book that shed some light on this. It's called the Epigenetics Revolution, and really well written. It covers that in depth. And when you asked the question, Ahmad, I was trying to think of how do I answer that question succinctly. In what what you read in this book, and a great example that's it's pretty well articulated in this book is in twins. So when you look at twins, they have the same genetic. These are twins that are genetically identical, versus the other kinds of twins that aren't genetically identical. So these are genetically identical twins, and yet they have differences, and those differences are not explained by the genetic sequence, but they are explained by the epigenetics.
Wow. Okay.
And when we have the capability to analyze a person's epigenome in a cost-effective way, in a rapid way, we can then create a personalized and precise treatment for
that
lifestyle
human.
Lifestyle intervention or something specifically for them. And I think that there's a tremendous opportunity, again, thinking about what are the specific problems that could be solved with an approach like that, and then how do you make it cost effective and really beneficial. And I think that's going to take a lot of work.
What's next for you guys at Inherent Bioscience? You got that SpermQT. What's next?
Yeah. So I talked about that situation where there's no sperm in the semen.
Right.
And we got excited about that because it's a big problem for these physicians and for the patients when they can't find any sperm. And so first, is there a way to help people start creating sperm that aren't creating it? And so understanding that, like that epi mutation that I talked about. The second thing that we can, pretty quickly and drastically help with is identifying if there's sperm in the testicle, so that those doctors and those patients can have success in doing the procedure. And so the way
what's the method to figure it out? Like currently, how are people doing it, and how could you do it better?
Yeah, so there's a couple things that people do. One is that they do a biopsy, fine needle biopsy. So they're poking the testicle and searching to see, and it's like mapping to see is there pockets of sperm. And obviously you can't check the whole testicle because that kind of defeats the purpose. The other is just to do it, just to fillet open the testicle, start looking. And a lot of times that's just what they do, and they start looking for it. So what we realized is that in the semen, there's biomarkers that can tell us whether or not there's sperm in the testicle.
Wow.
And one of those biomarkers is what's called cell-free DNA. So this is DNA that's outside of the cell. It's like when a cell dies, it releases all this DNA. And there's cell-free DNA in semen. And if it's from sperm, it's very unique epigenetically. And so on that DNA, we can look at the epigenetics and we can see, oh, this DNA came from sperm.
Okay.
If there's no cell-free DNA from sperm in the semen, there's probably no sperm in the testicle.
So don't do the other procedures. Oh, wow.
Yeah. And so we thought, okay, doctors aren't going to want this because it's going to reduce the number of surgeries that they do, the number of procedures they do. And we went and talked to a bunch of them. And that's the other key thing about entrepreneurship, is going and talking to, the people who experienced the problem and understanding it. And we asked them, we said, if we could do this for you, would you want it? Like, and how valuable would this be to you? And to our surprise, they said, oh my gosh, if you could tell me that would be so valuable, because the worst thing that happens to me in this case is that I do this procedure, I can't find any sperm, and I have to go out into the waiting room and tell, the female partner, I couldn't find any sperm.
Wow.
And then they get a bill for like $20,000 or $25,000
Wow.
for this procedure. And so they said, if I can at least tell them up front that there's a very small, I can still do this procedure, but there's no cell-free DNA from sperm in your semen.
Wow.
So I can still do this procedure, but we're probably not going to find any sperm. And, so that they are really excited about the test. So that's our next test that we're bringing out.
In the future, are you going to still, do you plan as Inherent Bioscience to stay focused on the male fertility problem, or do you plan to diverge into different problems that you might see in the future?
Yeah, so one of the things that we've realized working on this is that the semen is a really tremendous source of biological information and clinical information about men. Right. And it really has gone untapped, as a biological sample to understand the overall health of men. And it's kind of like a canary in the coal mine for men. If you have trouble, if you have things going on in your semen that can be detected, it can be an indication of other health problems.
Okay.
And so, like, one example is prostate cancer. Prostate cancer is the #2 cause of death in cancers of men. And we can see that same technology that we use for, is there sperm in the testicle for that procedure, that cell-free DNA technology, we can use that to understand what's happening with prostate health. And is there prostate cancer? Is it aggressive? Is it non-aggressive? And so there's a lot of things that we can do with semen that are really exciting. The technology that we leverage around epigenetics can be used in a number of different clinical areas beyond just men's reproductive health. But yeah, so we're excited about both the technology, but also very excited about the impact that we can have in reproductive health. And we've been focusing on men because there's been such a gap, but there's also an opportunity with female reproductive health in leveraging this kind of epigenetic analysis as well.
How big are you're selling both these, or you're planning on selling these products to the doctor, to the physicians themselves?
Yeah.
Or not to the customer as the patient, right?
Yeah. So you can order it directly through our website. We created a brand called pathfertility.com. So if you go to pathfertility.com, you can order the test. And what we do is we then get you looped in with a male fertility specialist who then can do a telehealth consultation and provide you with the results. And they also confirm that you're a candidate for the test, and they give us a physician ordered requisition. So it is a physician ordered test. And we started there because we want to get established in the clinical specialty and ultimately into the clinical guidelines. And then there's an opportunity to go directly to patients and give them access to this testing, directly, and market it directly to the patients. But we wanted to take the approach of working with the physicians first.
Because it honestly be a no-brainer for a 19-year-old, 20-year-old male to just do the test ahead of his years of planning on getting pregnant, and doing this test at a very early age,
checking it out,
making sure he's either yes, he could reproduce or not. If he can't, start working on the problem even before he thinks about it. That'd be a no-brainer.
Yeah, we definitely see that opportunity for direct to patient marketing, and we did some experimentation around that early on, because as a startup and as an entrepreneur, it's an iterative process. You want to find out, how do we get to a product market fit, and what's the market, and then how do we match up the product to it. And so we did a bunch of experimentation around direct to patient marketing, and we learned very quickly that it's very expensive, and it's a big educational burden. So we've talked, almost an hour,
and we could go. I could go for another 3 hours and more.
And when you start talking about the technology, there's a lot of kind of concepts that are new, and trying to explain that in a way that patients understand it is a challenge. And so when there's a physician and the physicians that we work with are highly educated, and they're really good at explaining things to patients, because they have to do that every day, all day. And so when we provide them with the information and the education, they then can translate that into, here's how I'm going to share that with my patient. And that has really accelerated, our initial market traction. And then as we learn, we can then say, okay, now we can market this directly to patients, and here's how we can do that and be effective, both from a cost standpoint and then also an experience standpoint that patient has.
Andy, I don't want to take a lot of your time. I could spend hours and hours and hours speaking about that, and I would like to have you in the future on another episode.
Sure.
Dive deep into different stuff. Where could people follow more of what you're doing? How could people find out more, and how could people help you in your journey? Feel free.
Okay, great. Thanks. Yeah, you can come to our website. It's inherentbio.com. And we picked the word inherent because we use what's inherent in our biology to remove the guesswork. So Inherent Biosciences, inherentbio.com. And then we also have our product website, pathfertility.com. And then also, I'd love to connect with you on LinkedIn. You can reach out on LinkedIn. We have both our company website or company account on LinkedIn, and then I'm also on LinkedIn. And help in the startup world and in the startup journey, it takes a village. So if anybody has suggestions, ideas, connections, introductions, that is the lifeblood of success in building something. And it takes a lot of collaboration and help. And so if anybody's interested in this area, wants to be involved or get educated about it, those are some great sources to go to.
Andy, thank you one more time.
Thanks, Ahmad.